Hello, good evening, everybody. Am I audible?
Yes, sir.
Okay, so let's start our today's discussion on medicinal chemistry, week 5 assignment, which I think you have already submitted. So today we will discuss about those questions. Okay, so let's start. Okay, is my screen visible?
Yes, sir.
Okay, so our today's first question is the following structure is called puromycin. What mode of action does it have? So now tell me, uh, what is puromycin? Anybody wants to say what is puromycin? Anybody?
Anti-cancer drug.
Not anti-cancer drug. Puromycin is a naturally occurring aminonucleoside antibiotic that inhibits protein synthesis by ribosome catalyzed incorporation into the C terminus of elongating nascent chains. Okay, so it has a very structural similarity with the aminonucleoside. Okay, so in this way it works. So it acts as a chain terminator in the protein synthesis. Puromycin is a naturally occurring aminonucleoside antibiotic that inhibits protein synthesis by ribosome catalyzed incorporation into the C terminus of elongating nascent chains, blocking further extension and resulting in premature termination of the translation. So, what is translation? What process is called What is replication, translation, and transcription? What is replication? Anybody?
Conversion of DNA into RNA.
Anybody else? Anybody else wants to say anything? Okay, the replication is not DNA to RNA. Replication is the process by which a double-stranded DNA molecule is copied to produce two identical DNA molecules. Means from one DNA, two identical DNA is forming. What is transcription? What is transcription? The previous somebody said the formation of RNA. Okay, from DNA, it is forming the one stranded, makes the RNA copy of a piece of the DNA. Okay, so it is the basically it is the formation of RNA. And what is translation?
Synthesis of protein from messenger RNA.
Exactly, synthesis of protein. So here in our this case, we are targeting this translation process. Okay, so let's see how does it work. Okay, so as I said earlier that it has some structural similarity with the aminonucleoside. So basically, our tyrosyl tRNA has the structure like this. Okay, so from here, this position, ester bond is involved. Okay, so from here, this is a amino acid. This part is an amino acid. Can you name this amino acid? This one. This is a very important amino acid of our body. What is the name? The phenyl ring and para position as OH group is there. What is the name of this amino acid? Okay, this is tyrosine, right? And from there we are getting this name tyrosyl tRNA. Means tyrosine is joined with this sugar moiety with this ester group, and in our puromycin molecule, the everything is identical. I am not drawing the whole thing, just the changing part I am drawing here. Here amide bond is there. Okay, the rest part is same. Okay, so as you can follow that here some oxygen, and if here oxygen is there, what we call this bond? An ester bond, right? And here the nitrogen is present, so we are calling it as the amide bond. So this is the structurally very much similar with the tyrosyl tRNA, and in this way it incorporates into the translation process uh, and hamper the synthesis of protein. So our right answer is, which is our right answer? It binds to a transport protein and prevents the reuptake of the serotonin. It binds to tubulin and prevents the formation of microtubule. It acts as a chain terminator in the protein synthesis. It acts as a protein-protein binding inhibitor. So which is the right answer? As we seen that what its action, it has a very similarity with this tyrosyl tRNA, and what does it do?
Terminate the protein synthesis.
Yes, it acts as a chain terminator in the protein synthesis. The third one. Okay, and very broadly I am showing that how does it do. Suppose here some tRNA is there, here tRNA, here tRNA, and here is peptidyl. Then after translocation, this is also tRNA, here, and this is the translation elongation process. And in presence of puromycin, here tRNA, here peptidyl is, and here puromycin is there. Then after translocation process, puromycin formed this bond with the peptidyl here, and the protein synthesis gets terminated. So in this way, it terminates the protein synthesis process. Okay, so let's move to our next question. So what is the molecular target of vinca alkaloids used as the anti-cancer drug? So now tell me, what is its target? Anybody?
So it actually prevent the synthesis of microtubule.
Hmm, hmm. So how it is used means what is its application in our medicinal chemistry approach? Yes, you are right. It stops the microtubule dynamics. That is why it is used in the cancer therapy, right? Okay, let's see. Okay, now at first we have to check what is, sorry, what is vinca alkaloid. Like the vinca alkaloid have two dimeric chemical structure. Okay, as I have already seen here from internet that it is a very complex structure, and composed of two basic multi-rings unit and indole nucleus. This one, this is an indole nucleus, and dihydroindole nucleus. Here a dihydroindole nucleus is also there. Here double bond is absent in the indole moiety, so it is called dihydroindole nucleus joined together with other complex system. Okay, now tubulin is the biochemical target for several clinically used anti-cancer drugs, including paclitaxel and the vinca alkaloid, vincitrin, and many other drugs. A vinca alkaloid used to treat breast cancer, testicular cancer, neuroblastoma, and lymphoma, mycosis, fungosis, and many other diseases. A vinca alkaloid derived from the vinblastine used for various types of the malignancies, but mainly acute lymphocytic leukemia. The mechanism of action of the vinca alkaloid is that the inhibition of the polymerization of the tubulin microtubules. The cellular protein tubulin, which occurs in the alpha and the beta form, it has the two dimer alpha and the beta, is essential for proper cellular function. During mitosis, tubulin polymerizes from the microtubules, okay, which are long tube-shaped protein polymers. The equilibrium between alpha and the beta tubulin and the microtubules is an important one. Of any disruption of this equilibrium can send dividing cells into the mitotic blood apoptosis. The vinca alkaloid binds to the beta tubulin at the different site from the paclitaxel taxol binding site and act to prevent tubulin assembly. Okay, so can you tell me what is microtubule in one sentence, very briefly? What is microtubule and what it is function? Okay, microtubule is the cytoskeleton protein. Okay, so it keeps the cellular shape, deformity, and integrity of the cell. And this is very important protein, microtubule, and it has the two isoform, alpha and the beta, and tubulin is polymerizes to the microtubule unit. Okay, so in what happens in cancer, as what is the characteristics of a cancer cell? The first very known characteristics. How do we recognize cancer cell? What is its means very fast criteria? Anybody?
So metastasis.
Metastasis and uncontrolled cell growth, right? The cell growth is very much in an uncontrolled way, right? So what happens in cancer when tubulin is polymerized to tubulin is polymerized to microtubule in a very fast matter. So what our alkaloid does here, I have shown. Can you please follow? This is the microtubule. This is the polymerized state, right? So this is the state in the what we call say in the cancer. It is very much polymerized or it is divided manner. What does vinca alkaloid do when we put this alkaloid in this system? It is enhancing the depolymerization. Can you see here the vinca alkaloid is taking place in between the tubulins, and the polymerized or well mannered shape is disrupted here, right? So break down the assembly of this tubulin. So in this way vinca alkaloid works. So the tubulin depolymerization takes place, and this polymerization and depolymerization of the tubulin is a very dynamic process, and there should be a proper balance to maintain our daily normal body physiological processes. So our right answer is here tubulin, right? The others are wrong. The transport protein for serotonin, DNA, or cell membrane. So our target for the anti-cancer drug is tubulin. Okay. Can I move to the next question?
Yes, sir.
Okay. Okay. So here a big structure is given. So which of the following statement is true regarding the amphotericin? Now what is this? What is this amphotericin?
Yes, antifungal drug.
Yes, it is an antifungal medication used for serious fungal infection. Okay, so we have to check. This is the structure of this amphotericin. So we have to say what is the true regarding the amphotericin, right? So now we are coming one by one. So now first one is saying the blue region is a hydrophobic region that interacts with the phospholipid bilayer of the cell membrane by van der Waals interaction. So is this true or false? So what we can say here, in the blue region a conjugated double bond is there. So is this part hydrophobic or hydrophilic? This part hydrophobic region or hydrophilic region? Hydrophobic region, exactly. No such polar group is there. Only alkene moiety is there, conjugated double bond. So definitely it is the hydrophobic part. So is this possible? The is the hydrophilic? What it is written here? The blue region is hydrophilic region. So is this true or false?
False, sir. It is a hydrophobic region.
Yes, it is false. So it is, we are eliminating out this option. So the next option is several molecules of amphotericin self-assemble to form a hydrophobic tunnel through the cell membrane. The tunnel interior is lined with nonpolar functional group. So is this true or false? Can you see a big tunnel is there? The molecule itself forming a tunnel type molecule. A cavity is there and the whole molecular structure is surrounded around it. So this cavity is hydrophobic or hydrophilic?
Hydrophilic.
Hydrophilic in nature.
Hydrophilic, exactly. Some OH group is there in the tunnel. So definitely the tunnel is hydrophilic in nature. Okay, so this is also false. To form the hydrophobic tunnel, this is not true. Okay, so the last one, what we are saying, that amphotericin act as an antifungal drug by allowing the uncontrolled passage of the ions and water across the membrane. So this is the right option. C is also false, since in C we are saying the amphotericin molecule is the ideal length to span the cell membrane. C is false, since amphotericin is only long enough to span the half width of the cell membrane, and so the tunnel involves two clusters of the amphotericin molecule arranged end to end. So we are getting the A, B, C are the false. Then only answer is the option D. Okay, so what is this structure? The following structure is used in the treatment of the brain tumors. So what is this structure called? What is the common functional group in this structure? Double bond O is there and some NH is there. Then N, double bond O, and some subsequent is there. So what we call this, that NHCO NH group, what do we call this moiety? Anybody? This part, what we call this part?
Diamide.
Yes, diamide we can say. We call it urea,
It's a
right?
huh?
It is a derivative of urea. And
Urea.
here
Right.
also nitroso group is there. So we will call it nitrosourea, right?
Yes, sir.
Okay. So now we have to say that what kind of structure is this? Okay, so it is carmustine or lomustine or streptomycin or cyclophosphamide. Which kind of structure is this? This,
Lo-
sorry,
Lomustine.
lomustine. Yes. So all these kind of structures are used in the brain tumor to treat brain tumor. Like, so what is okay? Yeah, this is the lomustine. Now let's see that what other kind of action of the other kind of drug. See, carmustine causes cross links in DNA and RNA, leading to the inhibition of DNA synthesis, RNA production, and RNA translation. Carmustine injection is used to treat certain types of the brain tumor. So what is the structure of carmustine? It is a very similar like structure only. This is the structure of carmustine. What the function? How does it work? It cross links in DNA and RNA. Okay, so here the streptozotocin is a diabetogenic agent extensively used to induce diabetes and to study complications inducing diabetic peripheral neuropathy. Okay, this is the function of the streptozotocin. And the last one, that is the cyclophosphamide, it works by damaging the cell's DNA and may kill cancer cell. It is a class of the medications called alkylating agent. It works by damaging the cell's DNA and may kill cancer cells. It may also lowers the body's immune response. So this is the working principle of the cyclophosphamide. And our desired one, that is this structure, is the lomustine acts by slowing this process down. It kills cancer cell by damaging the DNA and stops them from dividing. Lomustine is a highly lipophilic nitriurea compound which undergoes hydrolysis in vivo to form the reactive metabolites. Okay, so this structure matches with the lomustine. So this is the option B is the right answer. Okay, so in our next question, what is the mode of action for the anthracycline anti-cancer agents? So what are the common mechanism of these anti-cancer agents. They may be a alkylating agent, they may be a chain cutting agents, or antisense agent, or intercalating agents. So now we will go one by one in short. What are the classification of these agents? How does it work? And we will see that what is the mode of function of the anthracycline anti-cancer agent. Okay, so first we will discuss about the alkylating agent. So can anybody tell me what is the function or how does it work, this alkylating agent? Anybody? Okay, so alkylating agents, what is its common mechanism of action? Alkylating agents are a family of the anti-cancer drug. Okay, as I have said earlier, that all of these are the anti-cancer drugs, and anti-cancer drug that interfere with cells' DNA and inhibit cancer cell growth. They are so named because of their ability to add alkyl groups to negatively charged groups on the biological molecules such as DNA and proteins. So what does it do? They alkylate the negatively charged groups like DNA and the proteins. They have some polarized functionality. So the alkylating agent, these alkylating agents add some alkyl group to this DNA and the protein molecule, and so they cannot form their normal transcription, translation, or replication process. In this way, they forms the cross-linking the DNA strand, inhibiting replication and transcription of the RNA. So this is their mechanism of action. Some common alkylating group is can work like this way. As you can see that here a alkyl group CH3 is present in the nitrogen. So what does it do when it interacts with some negatively charged DNA or the protein molecule? It transfers these groups to the DNA or protein. In this way, they hamper their normal division process. In this way, they blocks the cancer cell or cancer mechanism. Okay, now the second one is the chain cutting agents. Chain cutting agents, from the name, can you guess the function of this uh, mechanism of this action? Anybody? From chain cutting name itself, what you can guess from this name?
Diradical formation.
Yes, exactly. From the name itself, we can guess or assume that the chain cutting agent generated DNA diradical. Diradical reacts with the oxygen and results in chain cutting. Okay, so in this way the diradical formation is happen. Then this diradical reacts with the oxygen present in the cell, and it results in the cutting or cutting down of the chain. Then the third one is the antisense agent. Target inhibits the transcription. So how does it work, the antisense agents? What is sense and antisense?
It does not respond.
Mhm. The antisense means the, as you know, the two strands of the DNA. If one is 3 prime to 5 prime, then another is, and what we call this one? If it starts from the 5 prime to 3 prime, then the another one is
3 prime to 5 prime.
3 prime to 5 prime. Then one strand we call sense and another one we call the antisense one, right? So how does it work? If we add a chemical compound which have some complementary sequence of this one of the strands, see here some group is there, then our antisense agent has some sequence complementary to this one. Then what does this compound do? It comes in between these two chain and breaks the normal helicity of our DNA and break down the transcription process. It is chemically synthesized. We are putting intentionally to stop the replication and the transcription of the DNA and the RNA. So we are intentionally putting this chemical structure within this structure to stop the transcription process, to inhibit the cancer mechanism. Okay, so these are the three strategy, and the last one is the intercalating agents. Now you tell me how does it work, the intercalating agents? Anybody? Okay, the intercalating agents is the one that directly interacts with the DNA. Like for example, here a DNA is formed. Here is the two strands of the DNA. Here A, here C, here G, here C. So here T, here G, here C, and here G. This is the base pair, and here some phosphate sugar backbone is there. So some, what is the intercalating agent? Some with some spacer, it directly interacts with the DNA molecule. This is the chromophore 1. This is joined with this spacer, and this spacer there is another one, that is chromophore 2. So with the linked up with this spacer, it has some groups which can directly interact with the DNA molecule, and it comes in between the base pairs and interacts with them. So in this way, intercalating agents operate or react with the DNA and stops the normal transcription and the translation process. So what is the example of this anthracycline anti-cancer drug? What class does it belongs to? This belongs to our last one, that is the intercalating agents. It directly interacts with the DNA and stops the cancer growth. Okay, so now let's move to our next question. Okay, so here some elaboration I have put of the intercalating agents that contain planar aromatic or the heteroaromatic ring systems. Planar systems slip between the layers of the nucleic acid pairs and disrupt the shape of the helix. Preference is often shown for the minor or the major group. Intercalation prevents replication and the transcription. This is very important. That why does it use to prevents replication and transcription. So intercalation inhibits topoisomerase 2, an enzyme that relieves relieves the strain of the DNA helix by temporarily cleaving the DNA chain and crossing an intact strand through the broken strand. So intercalating agents 2, here is some action. You can go through this. I think this will help you in understanding the intercalating agent mechanism. So coming to our next question, that diminished delivery of the oxygen to tissue is termed as, this is very easy, you should answer this.
Hypoxia.
Hypoxia, exactly, very good. So this is hypoxia. So I have also noted that what is the other term means in very short, that what is ischemia. What does ischemia means?
So
Okay,
when the blood supply to some part or tissue is
yes.
uh,
Yes, absolutely right. Ischemia is the condition in which the blood flow is restricted or reduced in a part of the body. Okay, this is ischemia. And what is homeostasis? Homeostasis is the state of the steady internal chemical and the physical condition maintained by living systems. This is homeostasis. And metabolism. Metabolism is the state of the life-sustaining chemical reactions in organisms. The three main functions of the metabolism are to conservation the energy in the food, to energy available to run the cellular process. Means all the chemical reaction which always takes place in our body, that is we call a metabolism. And last, what is hypoxia? That is our desired answer here. Hypoxia is a state in which oxygen is not available in sufficient amount at the tissue level to maintain adequate homeostasis. This can result from inadequate oxygen delivery to the tissue either due to low blood supply or low oxygen content in the blood. Okay, so this is the hypoxia. Reduced, diminished delivery of the oxygen to the tissue is termed as hypoxia. Okay, so here the following antibiotic structure is an example of the intercalating agent. So we have already discussed about the mechanism of the intercalating agents. What role does the ionic group serve in making this molecule an effective intercalating agents? So what does intercalating agents do? What does intercalating agents do?
Interacts with nitrogenous bases within DNA molecule.
Yes, basically it interacts with the DNA molecule directly, right? Interacts with DNA directly. It may be through the nitrogen base pairs, it can form nitrogen base pairs, or maybe via through some other groups. So what we are supposed to answer here, that this amine polar group that is present here, this ionic group, serve in making this molecule effective in intercalating agent. Okay, the first one is it increases water solubility, allowing high dose level to be used. So is this true or false? Keep in mind that we are thinking in terms of this term intercalating agent. Okay, so in terms of the intercalating agent, is this true or false? It interacts, it increases water solubility, allowing high dose levels to be used.
It is wrong.
This is wrong, because in that sense it is true that this polar group increases the solubility, but it is not true with respect to our desired mode of action, that is the intercalating agent. Okay, so if this is true, this statement in a way is true, that it increases water solubility. This amine tertiary amine group is very much polar group, and it increases the water solubility, but it has no effect, means this solubility has no effect in terms of the intercalation. So in this question we are marking this as a wrong one. Okay, next in C it is saying that it acts as a strong hydrogen bond donor with nucleic acid bases to stabilize intercalation. Is this true? This one, C, it acts as a strong hydrogen bond donor with nucleic acid bases to stabilize interaction. No, this is not. A strong hydrogen bond donor has nothing to do with this intercalating agent. Okay, so this is also false. It only becomes potent when it enters cells and so becomes trapped within the cell, leading to high concentration of drug. So is this our criteria for intercalating agents, that it gets trapped within the cell and have a high concentration of the drug? No, this is also false. So our the last option which is left, that it interacts with the charged phosphate group in DNA to stabilize the interaction. So as we said, that it can directly interact with the DNA molecule. It has some group which can interact with the chemical reactive active groups of the DNA. So This ionic group interacts with the charged phosphate group, that is of the sugar phosphate backbone of the DNA molecule, and stabilize the interaction. So it, option B is the right answer. It reacts with the phosphate group and stabilize the interaction. So this is the right one. Okay, now answer this question. Which of the following carries acyl group in thioester linkage? Is this acyl carrier protein or acetyl-CoA ACP trans-acetylase or enoyl ACP reductase or malonyl-CoA ACP transferase?
Option A is correct.
Option A is correct, acyl carrier protein. Okay, acyl carrier proteins (ACPs) usually have 70 to 100, but occasionally more amino acid residues, and they are usually linked through the interior serine residue to the terminal phosphate group of the 4' phosphopentothioane phosphatic group. Beta-ketoacyl ACP synthase condenses acyl and the malonyl groups. Malonyl-CoA ACP transfers malonyl group from the CoA to ACP. Okay, so option A is the answer. Now the following antibacterial agent was used in the Second World War. What is the name of the structure? What is the name of the structure?
Proflavin.
Proflavin. And what is the mechanism of its action? We have already discussed several ways today.
Intercalating agent.
Yes, intercalating agent. It interacts directly with the DNA and thereby disrupting DNA synthesis and leading to a high levels of mutation in the copied DNA strand. This prevents bacterial reproduction. So it is very much widely used in case after the Second World War to prevent the bacterial infection. Okay. Okay. So here what we have, dihydroxyacetone phosphate is rapidly and reversibly converted to which one? Dihydroxyacetone phosphate DHAP is converted to G3P.
Glyceraldehyde triphosphate.
Yes, glyceraldehyde diphosphate by the enzyme triose phosphate isomerase. As the name suggests, this enzyme catalyzes the isomerization of the 3-carbon sugar into another 3-carbon sugar. Here I have, sorry, here I have noted down this mechanism. This is the D-glyceraldehyde 3-phosphate G3P, which is the our option A, and this is rapidly converted to the dihydroxyacetone phosphate via the enzyme. This is basically isomerization reaction, and this is gone through the enzyme triose phosphate. So what is the basic isomerism happens here? What is group present here in case of the G3P?
Aldehyde group.
Yes, this aldehyde group is present here. And here, what group is present?
Ketone.
Phenol.
Sorry,
The ketone group is present.
yes. This is not phenol, okay? Phenol is when the OH group
Ethanol.
is linked with the phenyl group, okay?
CH.
This is alcohol group, and here the isomerization takes place. The keto group is formed. Here some aldehyde group was there, and here ketone group is there. Okay, clear? Okay. Okay, now let's move to our next question. Okay, this is very easy. The following agent is used for the treatment of the testicular and ovarious cancer. What does it compound called? It is very common.
Cisplatin.
Cisplatin. This is cisplatin. And what it is action? Okay, the following agent is used for the treatment of which of the following statement is untrue regarding the above statement? Okay, so here the chlorine groups are displaced before the drug becomes active. This is true or false? And how does it work? First, tell me. This is a very common type of the cisplatin, and it forms some chelating agents with in the presence of the DNA, and we have to point out which of the following is the untrue regarding this cisplatin. So the chlorine groups are displaced before the drug becomes active. Is this true or false?
True statement.
True statement. It is true. Is it true? The first statement is very true because when we put cisplatin in our cell, then this is a cisplatin, then it takes place, hydrolysis is takes place. Hydrolysis is takes place, and one of the chlorine is displaced by the water molecule. Okay, so our first statement is true. Right. So yes, for its action, it has to form some displacement of the chlorine has to takes place before its action, and some water molecule comes into the place of the chlorine. So the first option is true. So now the second one is intrastrand cross-linking of the DNA results from the action of this agent. So is this true or false? What I have said earlier, that with DNA, if this is DNA, it forms a direct interaction with the DNA. So the second option is true or false?
True, sir.
True. Second option is also true. The intestinal cross-linking takes place in case of the cisplatin. And the last one, the compound acts as the metalliting agent. Is this true or false?
Yes, it's a metallic image.
Yes, this is also true. It acts as the metalliting agent. Uh,
That
only the only the C1, that base pairing between adenine and thymine, is disrupted by this agent. This is the false one. It is the base pairing between the cytosine and guanine which is get disrupted. Okay, here AT is there in the question, but in case of the G and C, the disruption takes place. Okay, so only the option C is untrue regarding the cisplatin. Okay,
yes, sir.
so let's move to the next question. So now tell me, pyridoxal phosphate and its aminate form pyridoxamine phosphate are tightly bound coenzyme of aminotransferase or glutaminase or glutamine synthase or glutamate dehydrogenase.
Aminotransferase.
Aminotransferase. Yes. So pyridoxal phosphate is a coenzyme of the amino acid metabolism. In some of its reactions, which are transamination, it acts as the amino carrier. Pyridoxal phosphate is an excellent example of the single coenzyme capable of catalyzing complete different reaction. Okay, so the right option is aminotransferase one. Okay, so now metabolic intermediates found in the living system which are essential for the growth and life is called the
Primary metabolites.
primary metabolites. What is primary metabolites?
Carbohydrates.
Okay, and any other? Any other primary metabolites?
Glucose.
And what is the general form of glucose? Glucose is a particular form of that substance. Glucose is what? It's basically a carbohydrate,
Yes, sir.
right? There are four main primary metabolites, that is carbohydrate, protein, lipid, and vitamin. Okay, it's as we know that they are very essential for our primary one. And what is secondary metabolites? What is the example? Some kind of example and can you give of secondary metabolite? Okay, secondary metabolites are the some other specialized metabolites like toxin, okay, secondary products or natural products or organic compounds produced by any kind of life form like bacteria, fungi, animals or plants. Like the natural product, this is the secondary metabolites of the plant, which they make them competitive in their own environment. Okay, this is the secondary metabolites, and our desired answer is the primary metabolites, are the essential metabolic intermediates of the plants which help in the growth and development of the plant. This is in case of the plant, and in case of the animal, we have the as we have discussed that carbohydrate, protein, fat. This is the primary metabolites. Okay, so what are the main enzyme system for catalyzing ethanol to acetaldehyde? Alcohol dehydrogenase catalase, alcohol dehydrogenase cytochrome P catalase, and cytochrome P. What are the main enzyme for ethanol to acetaldehyde?
Alcohol dehydrogenase.
Alcohol dehydrogenase. So, what is the structure of the ethanol?
CH2OH.
Hmm. And the acetaldehyde?
CH3CHO.
Yes, yes. Right. So here, as we can, it is a what kind of process is taking place? Reduction or oxidation? From ethanol to acetaldehyde? So oxidation or
Reduction.
reduction?
Reduction process.
Reduction, sir.
Ethanol to acetaldehyde.
Oxidation, sorry.
Oxidation.
Information. Oxidation.
How that reduction can take place? Here hydrogen is eliminating out, and here double bond is forming. From here this hydrogen is
In,
eliminating
in
out, and
by
from here one hydrogen is eliminating out, and this C double bond O bond is forming here. So elimination of hydrogen we know as it is a oxidation process.
oxidation.
Yes, and the main enzyme in this process is the alcohol dehydrogenase ADH. The alcohol dehydrogenase ADH pathway is the main pathway involved in alcohol metabolism, but then with continued alcohol consumption in the MEOS microsomal ethanol oxidizing system, it induced and becomes the dominant pathway. A small amount of the alcohol is metabolized by the catalase pathway. Okay, so our desired answer is alcohol dehydrogenase, by which the ethanol is transferred to the acetaldehyde. Okay, so we have ended up with our today's question answer session. So if you have, then we will meet in our next session. Okay.
Thank you, sir.
Okay, thank you for joining.
Thank you, sir.
See you in the next session.
Okay, sir.